All right, so our next speaker is Frank Erstrup from the Technical University of Denmark, and he's going to be presenting on opportunities and challenges with metagenomic-based wastewater surveillance.
Okay, thank you, and thank you for inviting me. And I was asked to talk a little bit about opportunities and challenges with the metagenomic-based wastewater surveillance. And I think that it's really super to see how wastewater surveillance has taken off, especially after the COVID-19. And I think that what we see is, at least from my side, a lot of these, single target surveillance programs, which definitely have their challenges of their own. What we have been working with has been a more agnostic approach, where we just go out and try to use metagenomics to do surveillance for everything. Our focus has been antimicrobial resistant genes, which has been more than sufficient for us. But I do think that it has waste opportunities, and it basically can be a method for doing surveillance of everything. Yeah. So, and I think that I probably don't need to convince you that we basically need surveillance of everything, and that is really the key for any kind of public health interventions. No matter whether it is a question of going out and doing treatment, if it's a question of choosing the right vaccines. It's a question of, sending people to sub-Saharan Africa. Well, you need to be able to know where to go and what you're going to be up against. And it would really be wonderful if we could get this global surveillance data sharing up and running. And being one that's been working for this for quite a number of years, especially together with the World Health Organization, I think that the advantage of next generation sequencing is really something that we should really embrace, especially because of the common language that we have. That raw data can be actually reanalyzed, which if I understand the previous presentation correctly, was also the case that the discovery of the new variant was actually not in the wastewater, but it was reanalyzing afterwards. But perhaps you can correct me afterwards. But then especially that these laboratories, especially in low middle income countries that do not have the full setup, can really leapfrog, especially now with the new mobile sequencing technologies coming on board. As I said, we've been focused on metagenomics, that is, just going out, taking the samples and randomly sequencing everything. It does come with a number of different challenges that actually increase if you start thinking over it. It is really important that you do a purification of the DNA that is unbiased towards what is in your sample, so you do not suddenly go out and use a method that are only purifying gram-negative, gram-positive, or certain single-stranded RNA viruses versus double-stranded and those kind of things. So it's really something that requires a lot of standardization, especially if you want to have a high reproducibility. And then, as I also said, wastewater has been a focus area from our side, perhaps mainly because I come from a technical university, and we do not have the advantage of having access to a clinic where we have these nice study objects that are called patients, and you can get all these nice convenient sampling. So we had to look elsewhere. I also think, of course, that it makes a bit of sense. I've been part of antimicrobial resistance surveillance in Denmark for 25 years and was part of setting up the system here. And what we are really missing for a long time has been the healthy population, whereas hospitals, animals, livestock, food, we can get those samples, but the healthy human population is very, very difficult to do surveillance of, even in a super controlled country like Denmark. So those that might be carriers of infectious disease, which we think wastewater was really an ideal opportunity. For me, it started in 2014 when we decided that, okay, perhaps this is time to go out and see, is this something we can even handle, and can we even utilize metagenomics? And well, today I think everybody can do this, but just 10 years ago, it was perhaps a little bit more state of the art than it is today. At that time, we thought that we could look at long distance airplanes, and you have these 400 liters of brownish water that you can take off an airplane after 12 hours flight from that was from North America, as well as North and South Asia, and then simply flying into to Copenhagen, doing sequencing and doing read mapping to a reference database of antimicrobial resistant genes. And I hope that I can convince you that there was less resistance here in the green bars. That was North America. Well, that's main USA. There was one flight from Toronto, and then there's most resistance genes in the flights coming from Southeast Asia. Kind of made sense in terms of what we imagine. And also, this could be used to look for viruses, in this case, diversity, where I think you can also see that there's simply a different diversity of viruses when you come in. At that time, we never even thought of looking for, SARS-CoV-2. It didn't exist at that time. We also thought that it could be something that might be useful in a low middle income countries, and doing sampling here in a slum city in Kibera in Kenya, and is simply following over time the different viruses. So I think that at least as a proof of concept, it seemed to be feasible. But the interpretation and also how to use these results, especially in such a setting with 250,000 people living in the biggest slum area in the world, that certainly proved out to be a little bit more complicated. Then, fortunately, COVID-19, happens, and we didn't have to bother with how we should, implement these kind of surveillance in a real life setting, because now I think it is done overall all over the world. So one of our focus areas for the last several years has been, to test out a global surveillance for antimicrobial resistance based on urban sewage. And in 2016, we reached out and tried to see if we could collect samples from all over the world, and in this case, 79 cities, 63 countries, did sequencing, and looked at the abundance and also the basic diversity across the world. And as you can see, most resistance in Africa, and the lowest level of resistance mainly here in Europe, perhaps Oceania, that's Australia and New Zealand. But at least to our surprise at that time, we really saw this drastic separation in the diversity of the resistant genes that was found across the world. That has puzzled us. It is still puzzling us a bit. The resistant genes we have looked at in that case was 3,100 genes that we know has been mobilized from their original context. So this is what we call acquired resistant genes. But nonetheless, this is what we've seen, and I think that we reported that we saw this clear separation. We then did a follow-up study a couple of years later, where we just continued to do the sampling. In that case, quite an additional number of cities and 101 countries across the world, and clearly highest abundance here in sub-Saharan Africa, but also Peru and Brazil, which were pretty high abundances, and then lower abundance further up north and further down south. So almost like a equatorial abundance. Diversity, we could still confirm a little bit more muddy pattern, but could still confirm that we see this separation of the world's regions in different ways, but also depending on the classes of antimicrobial resistant genes, it actually tends to differ. One of the things that we were particularly intrigued about was that when we actually started to not only look at the present absence of the different resistant genes, but they looked a little bit more into details. So that is that if you have a resistant gene, you can also look at the exact variant of that resistant gene. So if it has some point mutation, so a little bit like SARS-CoV, you can start looking also at the variants of these genes, and then also at the flanking region, so the gene synthesis, what is the context in which it's sitting. And at least for the 49 most common resistant genes we saw, we were able to do a gene synthesis, looking at variants. In this case, it is which bacterial host we expected this is in, and then also with the regionalism. And in this case, the red is Africa. I think perhaps I can convince you that we do see, in some cases, a pattern that certain variants are more common in one region compared to the other regions. And this was something that we wanted to follow up with, in something that is still unpublished. This is based on 1,240 sewage samples from 351 cities in 111 countries around the world, collected between 2016 and 2022. We still see, as we have seen previously, this separation. Now we can have more dots, so it starts to get a bit more muddy, but we still see some kind of a regional separation when we look at the present absence of the resistant genes. But perhaps more for us, a little bit more intriguing, that is, if we then go in and actually do metagenomic assemblies of everything, and then look at what is the exact variance we find. I think that is probably the predominant variant, and not, a mix of variants as for the SARS-CoV-2 in the previous presentation. But then if you look at the different variants, well, whether the different cities, do we find the same variants, or do we find different variants of it? In this case, 14,040 different resistant genes, both those that have been acquired, that's on the top, and those that have only been identified through functional cloning. And well, you can debate what you want. This is just geographical distance, and then the dissimilarity plot. But at least to us, it looks like we see an increased dissimilarity if we have an increased distance between the cities. So simply, a distance decay of the variant. So basically, you have a variant popping up, and then it picks up more mutations as it gets further and further away. I don't think we will be able to see these kind of patterns if we just looked at single genes. But when you look at several thousands of genes, then at least it seems to be picking up like this. The red color is if it's from different dots from different regions, and the blue is if it's in the same region. And in that case, we actually don't think that we are seeing a separation based on these artificial defined regions we have, but simply a question of how distant cities are from each other. We cannot see any impact at looking at flight connections. My guess is that number of flight connections, the number of people that are moving through flights is simply too limited to really make an impact on these kind of patterns. But it remains to be seen, and if someone else wants to have a look at the data and tell me that we are totally wrong, please go ahead. So we have also been doing in-depth study in 5 European cities. So that has been Bologna, Budapest, Rotterdam, Rome, and Copenhagen. And we had 3 sites in Copenhagen where we have been sampling over more than a year. One of the things we see that is that we can see a very, very, very strong signature in the both the resistome but also the bacterial composition of the different cities. So basically, when we have more than 10 samples from a sewage treatment plant, I would basically claim that we can tell. Then number 11, we can tell exactly what sewage treatment plant that was coming from. So really can get these very, very, very specific signatures. So at some point in time, we might not even have to collect metadata about what is happening, where the samples come from. Another thing is also that we definitely do see some temporal trends over time, so simply annual trends and other things that we believe could be important in the future, also to tell something, to get information about exactly what time of the year was the sample from, and then account for this. But for us, perhaps a little bit more intriguing is that when we start having this large number of samples, we can also do network analysis. And I think that there was a question to the previous talk where we talked about okay, what if there's different kind of source contaminations into the sewage system? Well, then how do you actually relate this that something might be coming from a different source? In this case, we do believe that we can start getting clusters of fecal bacteria and simply say, okay, well, they are co-abundant together. And then we know that some of the bacteria we know are fecal, but then we can also assign all these other bacteria that have not yet been assigned as fecal bacteria that seem to be co-abundant with the fecal bacteria. So we can simply assign, this fraction is the fecal contamination in sewage. And then we can also start assigning, well, how are the resistant genes linked to this? And then use this for surveillance in the future. I definitely think it's something that would need more work in the future, but nonetheless, I do think that this is also where the technologies can get us, and also getting these more advanced modelings. Sewage-based surveillance, as with anything else, is something that is highly dependent on standardization. We have done, and actually from my side, just to complain, it is very difficult to get standardization and evaluations published. A little bit to my surprise, because I think that this is really what everything is resting on. But we have shown from our side at least that sample storage really matters. Do you freeze it? Do you not freeze it? What is the temperature? The DNA extraction, how the DNA is handled, how the sequencing platform, etc. All this actually includes, a bias in your result. Then also the downstream analysis, and I think that mapping to different resistant gene database, mapping to different bacterial databases, using different updates of the pipelines, etc. It is all influencing your end results, and thereby also the comparability of your results to others. However, if we are sharing the raw data, then all these dry lab methods actually might not matter, because then we will actually have the option to reanalyze and re-evaluate the data. So it's really a question of what we call the laboratory bias that is impacting this. So I'll just say that standardization is important. It is something that we need to take into consideration, especially when we're talking the determination of abundance. However, as we also see, when we do assemblies, then unstandardized data might still be useful for network analysis, and also work where we just want to know, do we have this or that variant in the different places? And then, of course, sharing of the raw data, and especially the raw data, not consensus data, will actually allow for multiple analysis. So, concluding remark, well, surveillance is the basis of everything, and without it, we are flying blind. It's very easy to say, but I do think that it has proven more difficult to conduct, especially on a global scale, than we have actually expected. Metagenomics and sewage offer several novel options for global surveillance. Standardization is important, but even data that are generated using different methods, different purification protocols, might still be useful for comparison. And then, of course, raw data sharing is essential and should really be mandatory any part of any funding. Thank you. So, any questions?
Can you hear me, Frank?
Yes, now I can.
Oh, awesome. Great. Just to kick off questions, I've got one. Really excellent presentation. Thank you so much. I was curious, when you look at the diversity of, across countries. So for example, the AMR resistance genes, and seeing the clustering by country and region. How do you incorporate it all the type of sewage, as far as, I can imagine in the US you have these, sort of very centralized sewer systems, and other places maybe you're sampling from like open drains, or, perhaps more contaminated, or lots of other things in the sewage. So I was just curious if that's a factor at all in your analyses, or if that's maybe a theory for some of the diversity differences. Yeah. Thanks.
Yeah. It actually seems that it has certainly some impact on the bacteria realm, but it seems to have less on the resistome. And also, especially, I think that with the network analysis, you can start extracting the sewer, or lateral pit or whatever associated microbiome, and then only look at the fecal contamination. But it is a factor. We do collect that data. We also collect data on how does it look, visual when we receive it, and have actually also just taken pictures, so people, if they want to have a look at it themselves, that is actually all should all be available. But it has not really proven as come up, as a major factor in our analysis compared to all the other factors. And I have to say, much to my surprise, because clearly there should be a different right, if you are in Moshi, Tanzania versus Copenhagen.
Fascinating. Thank you so much. We have one more question, I think, in the back.
Hi, thank you so much for a great presentation. I had a general question about the assemblies for the current work. How many are you getting any weird hybrid assemblies? Yeah, how often are you able to recover a lot of these AMR genes well? And are there any hypotheses as to why things might be doing so well or dropping out? Thank you.
Yeah. That's a good question. And the thing is, of course, you don't know whether you're getting hybrid assemblies right. Most of the assemblies we get actually map to known variants. I do think that we're getting some hybrid assemblies. I do think that we are missing out on some minority variants. But nonetheless, I do think in most cases we are probably getting the main variant that is, from that specific sampling site. But it's a good question, and I don't know. Perhaps that will be resolved later on when we get doing more long read sequencing, but that remains to be seen.
Thanks so much, Frank, for the great presentation and questions from everyone. Let's thank him one more time.